Good morning, dear students. Today we are moving to another section in the nucleus chapter. That is nuclear pore complex. It's a very important question for your examination. So normally, please don't skip this question, this part. It's very important. Okay. What is nuclear pore complex? Simply there are certain holes, perforations that is present in the nuclear membrane. Nuclear membrane is a membrane that cover the nucleus. Okay. Now what is the significance of this complex? It's very easy. We can clearly guess that. What is the role? As it is a hole, it helps for exporting and importing materials from nucleus to outside and also from outside towards the nucleus. That is the significance. Can you give some examples for those substances that is importing to the nucleus? Importing means that is carrying from cytoplasm towards the nucleus. Which all substances? Some examples. They are histones, RNA polymerases, DNA polymerases, gene regulatory proteins, RNA processing proteins. All these are required inside the nucleus. So, they all need to be imported towards the nucleus, and that import is taking place through the nuclear pore complex. That means that structure is very important. And give some examples for those molecules or compounds that is exporting. That includes tRNAs, mRNA, certain ribosomal subunits. All those are examples for substances that is exporting from nucleus to cytoplasm. Then what about the dimension? Its diameter is 120. Its diameter is 120 nanometer. And its mass is 125 million Dalton. And it is composed of 100 different proteins. 100 different proteins. Okay. And next we are going to understand that model of nuclear pore complex. Here is a picture that is showing the details in a cross-section view of that nuclear pore complex. First, I draw some schematic representation and explain to you. And for this nuclear pore complex, important terms we need to familiar with are outer cytoplasmic ring, inner nuclear ring, spoke ring, inner ring, central channel, cytoplasmic filament, and nuclear basket. After I explained to you that model, you will be familiar with all these terms. Okay, I will draw and explain the cross-sectional view of this nuclear pore complex. Okay, first this is the nuclear membrane. Only it's one part, and its center have that pore complex. And this is the outer nuclear membrane, and this is the inner nuclear membrane. Okay, and inside in this space, actually that nuclear pore complex is occupying. And that complex consists of two rings. An outer ring. I am drawing only half of that structure because it is a cross section. Outer ring and an inner ring. This is inner ring. Okay. And these two rings composed of 8 protein subunits. I just show you that 8 for managing the space. I am drawing like this. Okay, 8 subunits, because as it is a cross section, in one side I can show only 4. And similar to this outer ring, inner ring also composed of 8 subunits. Okay, 8 protein subunits. Clear? And from these two rings, spokes are arising. Spokes are arising. What do you mean by spokes? Is it clear to you? And these spokes emerge towards the center and form another ring, inner ring, a central channel. In this way, central channel it will form. And before going further, I am explaining more clearly what is spoke. Spoke means it is certain structures. In steering wheel, we can see spokes. Spokes are that radiating structure from that outer circular structure. These emerging radial structures are called spokes, and it come and meet together in the center. And coming to the case of nuclear pore complex, spokes are emerging from the outer ring and inner ring, and they come towards the center and form a central channel. Here that central channel is forming, and through that central channel, the compounds, proteins, whatever things are exporting and importing. Clear? Okay. So next, we need to understand about certain filaments. Filaments emerge from the outer ring and also from the inner ring. Outer ring is called outer cytoplasmic ring because it is facing the cytoplasmic side. And as it is this inner ring, as it is facing towards the nucleoplasm, it is called the nuclear ring, and from that ring also so many filaments emerge. What is the name of those filaments? Those filaments emerging from the cytoplasmic side is called the cytoplasmic filament, and the filaments that emerge from the inner nuclear ring are called the nuclear filaments. These are called the nuclear filaments, and these nuclear filaments come and join and form, they converge, they join together and form a basket-like structure towards the inner side of this complex, and that basket-like structure is called the nuclear basket. Nuclear basket. Clear? Then we can just go here and check whether all the terms is familiar to you now. Outer cytoplasmic ring is clear. I explained you. Inner nuclear ring. Spoke ring, here we have seen. Inner ring means inner channel is here. Its central channel is passing at the center through that inner channel. Then cytoplasmic filaments are those filaments that is emerging from the cytoplasmic ring, and nuclear filaments is not mentioned here anyway. Nuclear filaments are those filaments that is arising from the nuclear ring and they converge to form nuclear basket. Okay, then I hope that you all of you understand all the terms relating with this nuclear pore complex, and it is a detailed picture of that complex. Here you can see almost all those things. This is the nuclear envelope. This is a cytoplasmic ring. This is a nucleoplasmic ring. So the nuclear basket that is formed by the converge and joining of nuclear filaments. This is cytoplasmic filaments, and spokes here we can see, and that spokes comes towards the center and form another ring. It's called inner ring, and at the center there's a passage that is called central channel. Through that passage, substances are moving towards out and in. Okay. The mechanism of transport through this nuclear pore complex that we are going to see next. Two-way, the transport of materials, compounds takes place through the nuclear pore complex. That is, first method is passive diffusion, and the second method is an energy dependent transport, or we can tell it as a selective transport. Okay, passive diffusion itself means that it doesn't need any type of energy, and that transports is applicable to small molecules. Those small molecules moves towards the nucleus and also out of the nucleus through open aqueous channels. That doesn't need that nuclear pore complex for their transport because its size is small. So, its transport happens by the method of passive diffusion. Diffusion, we know that movement of molecules from a region of high concentration to low concentration without the requirement of energy. Next, coming to energy dependent. That is applicable to for macromolecules. That includes proteins and RNAs. And for their transport, it need the nuclear pore complex itself, and also that transport requires ATP. That means it is an energy dependent transport process. That transport occurs through the central channel. Central channel, we already learned that the central channel is present in the center of nuclear pore complex. Through that central channel is proteins and also RNAs are transported. Okay. Next, we are going to understand how the nuclear envelope is reconstituting as a cell undergoes cell division. So, during cell division, the nuclear envelope needs to dismantle to carrying out the whole division process. So, after that process, again that nuclear envelope need to reconstitute. How that process is taking place, that we are going to see here. We are starting with the prophase. Prophase is the first stage of cell division. Regarding more details of cell division, you will learn in the coming chapters. Anyway, now you just understand about something regarding the reconstitution of nuclear envelope during cell division. And it begins with the prophase. At the stage of prophase, the nucleus is existing in this form with the nuclear envelope, nuclear pores, DNA, nuclear lamina, everything is there, and that cell is going to start its cell division. So, nuclear envelope need to dismantle, dissociate. First, phosphorylation happens to that lamin. Lamin, we already learned that it is a building block of nuclear lamina, and that phosphorylation leads to the depolymerization of the nuclear membrane as a whole, and it dissociates and form in a discrete vesicle. I will show you that they dissociate and exist in the form of separate vesicles, that nuclear envelope. Okay, and that cell is going on through various stages of cell division now, and finally it reaches the last stage, that is the telophase stage. And on reaching the telophase stage, during its beginning phase, what happens? The phosphate group that is previously added to this lamins at the beginning of prophase, it is removed, and after dephosphorylation of the lamin, again the lamin reconstituted first, and that reconstituted lamins goes and bind with the nuclear envelope vesicles and leads to its constitution. In that way, by the end of, towards the end of cell division, the nuclear envelope again formed in that newly formed daughter cells. Okay, with this we can finish today's class and section. Thank you. Next class we will see about nucleoplasm. Okay.
I'm an icon, man. My girl about a 10. I should fuck her in a Benz.